Okay, move on. Okay, continue on the tertiary structure. Okay, so you have seen this alpha and beta plated sheet, how the protein is folded into secondary structure. Now you have to imagine how this secondary structure to form a tertiary structure to become more compact 3D. So basically the same interaction, mostly hydrogen bonding between the distant amino acid residue, which the formation of the tertiary structure will allow to form a globular shape. Right, so this is an example of chymotrypsin. So A, the upper part is the primary structure. So if we do a sequencing for the primary structure, so this is how the sequence looks like if we do a sequencing, right? So basically, as a whole, the chymotrypsin structure is like a three. This is the 3D structure where it's folded. Below you can see very clear the mixture of the beta beta alpha helix and the loop. All right. And then we look on the first one, alpha keratin. So this is a tertiary structure where you can find this alpha keratin in claws, fingernails, hair, and horns in mammals. So they are mostly composed alpha helix rich rod domains. Usually it's around 311 to 340 residues. So it will flank by non-helical N and C terminal domains. So you can see just now, it only have two, right? One N terminal and C terminal domains. Okay, so mostly they are linked together through disulfide bond that make up the folding. Alright, so this alpha keratin is more rigid, it is inextensible and insoluble. So the pairs of right-handed alpha helix will wrap and around each other to form a left twisted coil. Okay, so for example here, this is the keratin type 1 and keratin type 2. So you can see there is a N terminal, so the length will be different among different keratin. You can see the C terminal domain on the right hand side, and in the middle is the rod domains of the alpha helix, which you can see like a B here, right? So this is the alpha helix of the alpha keratin, and usually it will coil, right? These two alpha helix, and it can be become pair of coil like this. We call it protofilament, and lastly it become a filament, which contain 4 right-handed twisted protofibrils. So that's why it become very rigid, right? For example, like our nails and hair, all right? So this is to show you clearly how this alpha keratins is formed to make a hair from the alpha keratin twisted into left-handed superhelix that I showed you earlier. So this is actually one of the alpha keratin, which is made up of these protofibrils. When it's being combined together, become microfibril, and then become macrofibril. Later, it become a very big, become a spindle-shaped cell cortex. You see, so become that's why one hair or single hair become very rigid, although it's only a alpha keratins. All right, and then move on to fibroin and beta keratin. So you can find this in silk fibers, in spider webs, or in bird feathers. So it is also another type of fibrous protein. So it composed of stacked anti-parallel beta sheets. So previously is alpha. So this is mostly is anti-parallel beta sheet. So there are large stretches of this glycine. So you can see a lot of sequence of glycine in the structure of this beta carotene. So the glycine will all end up on one side of the sheet, and you can see another residue is alanine and serines. This is all non-polar, right? The first group of amino acid. And you will see that later this beta sheet will be packed, they are snuggling together. So the glycine surface to glycine surface, and the alanine serine will pack with the alanine serine surface. So this is what the statement mentioned. So you can see these are two different sequence, right? So the alanine and alanine will pack together here, right, between these two different sequence, and glycine will actually with the other glycine outside, right? Okay, and this is just to show you that the spider web is made up of beta sheet, antiparallel, and the to show you the position of the beta sheet surrounded by the alpha helix and beta bands. Okay, and also bird feathers is also made up of beta carotene. And then next is collagen. Still in the tertiary structure, it is a triple helix. So just now you see alpha helix. So this is triple helix. So this is also considered a fibrous protein, rigid and inextensible. So mostly you can see collagen in the connective tissue, for example like tendons, cartilage, bones, teeth, skin and blood vessels. So basically we consider it is as a tropocollagen because of the three intertwined polypeptide chain. So usually it is about 1000 amino acid in length. So it is a unique right-handed triple helix, which is there are three strands, right, which arrange in left-handed helical fashion. So mostly collagen will contain lysine, proline and hydroxyproline. So this sequence which make these collagen fibers are incapable of forming traditional alpha helix or beta sheets, right? So this is what does it mean. So when there is a poly means a sequence of polyglycine, proline and hydroxyproline. So it tends to become a sequence like a alpha helix, right? And then twisted. So even though it is actually a beta, but because of the sequence of the three amino acid, so it tends to tangle, become a triple helix, right? So that's why it become very rigid of this collagen-like structure. Okay, so this is how collagen looks like under a electron microscope, right? So if we look in depth inside of this electron microscope of this collagen, so the sequence, the packing of collagen molecules, right? So there is some gap between the, we call it whole zone of between the sequence of the uh, collagen, the twisted, we call it triple helix. Okay, and then now we move on to the quaternary structure. So we have seen several examples of tertiary structure. Now from the tertiary structure, it become more compact, and they can be 2 or 3 subunit. Minimum is 2 subunit, right, to form a tertiary structure, to form a quaternary structure. And the conformational possibility between these two subunit usually uh, also interact through non-covalent bond, right? Okay, so this is example of our hemoglobin, which have 4 subunit, 2 beta and 2 alpha. And basically the 2 subunit will have an interaction through hydrophobic region, or we call it as we consider as van der Waals interaction between two. So that's why it's very closely and tightly packed, even though it's not a covalent bond. right? In a protein structure, you also need to aware about this denaturation and renaturation. So when we do some treatment or for example during protein extraction or do some protein isolation, so we need to consider the possibility of denaturation or renaturation of the protein. So for example, if you heat or exposed to acid or high salt concentration, it will cause protein denaturation. Denaturation here means the protein structure, for example, that when it's bind or folded through cysteine or through hydrogen bonding, it will be unfold. So when it's unfold, it will lose their function, right? And this unfolding, it can be reversible depending on the treatment. You can cause the protein to be reversed. It can be folded depending on how you treat the protein. So for example here, this is the native state. So you know that quaternary structure, there are several subunit, right? Folded either through hydrogen bonding van der Waals. If you do treatment, it will open up the subunit and then it open up further, the disulfide bond become a denatured state. So this will cause the protein to lose its function and the active site of the protein also open up, right? So you can see example. So this is irreversible. If you do cooking, right, on the protein, we know that the egg white is uh, having high protein. We call it ovalbumin. So if you heat or you cook, you cause the protein to be denatured. You can see the protein now become precipitate, right? So it lost its function already. Okay. And in the lab, usually we can do a protein determination on the protein structure, but mostly this instrument is very high tech, and we don't have it in the faculty biotech. But you need to at least know what are the instruments that usually being used in the future if you would like to study or determine the protein structure. Okay, for example, primary you can do through Edman degradation. So usually this is to know the N and C terminal. But later the best is using a tandem mass spectrometry, or we call it peptide mapping. Alright. So the secondary structure you can do through this circular dichroism spectroscopy. So basically all the spectroscopy, NMR, the FTIR, and in silico prediction you can use to determine the secondary structure of the protein. While lastly, tertiary and quaternary, you can use X-ray crystallography. We have it in biotech, but since it's very high tech, so it's not, for undergraduate we are not allowed to do this experiment. Okay, but just to give you the idea what are the instruments that use, and NMR and electron microscopy, and also in silico. So in silico means you use software, right, to or computerized software to predict the tertiary or quaternary structure. Okay, to give you the idea, so later you have a samples, for example like fish, last time that you did in the lab, you extract lipid. So you can also extract protein, for example maybe from fish or from meat, right? So you know it's a mixture of many proteins that present in your samples. So you have to do a chemical through enzymatic digest to break up further the protein, right? Mixture to make it into a peptide, and then you have to run into an instrument, right? So usually we call it LCMS MS. So you will get the fragment of the peptide, and then from the sequence, then you will see, for example here, right? The match sequence, so it represents different protein. So there's a lot of protein in the samples, and then you can do a matching with a database that present in the system that link in the online databases, right? So just give you the rough idea. So if it's not in the databases, mean it is a unknown protein, right? So you need to do further analysis if it's an unknown protein. Okay. For the circular dichroism, so we don't have this in biotech. So it's help you to know the different what we call the conformation, either the structure having an alpha, beta conformation or random coil, right? And you can also see whether the structure of the different protein. For example, like here, A is thiophosphate isomerase, right? So based on the spectrum, so different protein may have different spectrum, right? So from the CD spectra, you will know there are at least 4 different proteins present. Okay? And FTIR, okay, is also giving you the idea of the structure of the amide, right? So here you know that there is 4 different part of the protein that you can observe. So from here it's just like a puzzle, then you have to predict the structure, right? So next is another instrument, 2D NMR. So that is 1D NMR and 2D NMR. For protein, it's not possible to determine the structure through the common NMR. So you have to use 2D NMR. So it can help you to see the distance between nearby atom, for example, like this structure, right? So it is not easy. So for students who are master in protein, can be able to determine this spectrum, right? Even I myself also not familiar with 2D NMR. So just to give you the rough idea how the data will be produced, and you have to do the data analysis. So there is a principle on how you can predict the structure. And then it's from after you do your prediction, like a puzzle, you see. So later you will know which amino acid link to which amino acid to come up with a conformation of the protein, right? And as I said, we have this X-ray crystallography in the faculty, right? So in order for you to know the structure, how big is the structure of, for example, enzyme, so you can crystallize the protein. We call it X-ray crystallography, right? So from the crystal, then you have to do a diffraction. So you will get a diffraction pattern. So this is also need to have the skill and the knowledge on how to interpret this diffraction pattern in order for you to know the structure of the protein based on the crystal. So they will be attached to a software where it come out with a electron density map like this, where you see that is an amino acid, you see, folding, because it's a structure, right? Meaning a protein being crystallized in their specific structure. Okay, so it maintain its structure, original structure and also function. Okay, so from the data of the uh, crystal structure of the protein, then you can know the using the in silico, using a software to reveal the modeling of the structure, either it's beta plated or alpha and so on. Okay, and just to share with you that this is very long ago that our team in the biotech. So I'm not sure whether you are aware or recognize, this is Professor Dr. Rajan Nozaliha. She actually already retired. So basically, I'm sure most of you know Dr. Sheikh Muzaffar Shukor, right? All of you recognize him?
Yes.
Okay, so he is actually an orthopedic surgeon, right? So he came to biotech during my time in. I joined the department 2009, right? So he during that time always come to the faculty to learn how to do this protein crystallography. So this team, Professor Dr. Rajan Nuzaliha, come up with a simple method which he will do in the space. So the sample that being brought up to the space is thermostable lipase and cold active lipase, which is actually they isolated from bacteria. Cold active basically they are isolated from Antarctica. Thermolipase, I'm not really sure where, but it's actually a lipase enzyme, right? That is thermostable, that can be commercialized. So Dr. Muzaffar Shukor bring the two samples to the space and do the simple method where he will turn the protein to crystal. So they want to see how the formation of the crystal in out of space, right? So this is just to share with you what is going on around the faculty. So, but this is long ago, right? I think 10 years ago. Okay. Okay, move on. So still can follow. So we have finished on the quaternary structure of the protein. So now we need to know some of the techniques that you really use to purify protein. So last Wednesday you do lipid extraction, right? So but in this but last week you just do a simple protein determination. You didn't do a protein extraction. But in the future, as I said, if you want to do a protein extraction and purify, so there is several steps that you need to consider. Okay, so these are three common steps that you really use to purify. Purify means to isolate it out the protein of interest. So for example, like you have a fish, there's a lot of protein, right, in the fish. So there is a specific enzyme or protein that you would like to isolate. Okay, so you have to do some steps. Okay, for example, here is you can isolate it out or separate the protein first based on their charge. So usually we use ion exchange chromatography. So there are two types, either cationic or anionic. So you will learn more this uh, basically if you do a enzymology, right? But you are not taking enzymology, right? This program, I think you just take principle of biochemistry, is it? Are you taking enzymology in the program for biotech?
No,
Okay,
I
so I
think
no, isn't it?
I
But
think it's in elective or something about minor.
okay, so if you later you want to learn more, basically we have a course on enzymology to do the enzyme purification. So, but this is just to give you the uh, what call this introduction only. So basically there are two types, cationic and anionic exchanger. So we know that protein have charge, right? So we can separate them according to their charges. So you can use ion exchange, and then you can also separate the protein according to their size. So you have to use matrix, specific matrix as the what we call this the stationary phase, so that it will separate according to their size of the matrix that you use. So meaning the bigger size of protein will come out first from the chromatography, right? And lastly is the common one that we use is immunoaffinity. So usually we will do step by step in order to get purified. So you can't do one step to get a purified protein. So usually we start with ion exchange followed by size exclusion chromatography, and then once we get partially purified, we have removed all the unwanted proteins, and then from 100 maybe like a protein, then less now become maybe 10 protein, then and now we can use immunoaffinity chromatography, right, in order for us to extract out the specific. So this is high selective capturing agent, where the matrix or the stationary phase, there is antibody that will specific to your protein. They will grabs or it will be selectively bind to the protein of interest, so that the rest will be removed out from the column. So then later you have to elute out your protein of interest. Okay, so I just give you just a simple idea. So this is when you do a protein purification. So you have to do like 5 steps. First, you have to do crude extract. So the same as I said, when you do crude extract, it is very important to take care of the condition, whether it need to be cool or room temperature. But usually protein, we will always keep it on ice, right? To keep the activity of the protein, because later you need to do some total enzyme activity, specific activity. If you lost the enzyme activity, you will not be able to determine, you see, whether the protein is present in the extract or not. So usually, as I said, when you do protein extraction, even your sample, for example, if you extract from fish, you have you have to make sure the fish is on ice, right? So you have to extract out. If for example you extract from the brain to get the acetylcholinesterase, for example, so make sure the brain is on ice as well after you remove out from the fish. Okay, once you do the extraction, then you have to do salt precipitation, right? So this is based on solubility of the protein. Okay, and then continue with ion exchange. You do a molecular sieve, chromatography, and lastly immunoaffinity. So this is the step. So as I said, you will learn more if you take enzymology. So you will see the pattern. When you do a protein purification, if you trace, you see, of course the crude extract will be the highest, right? The highest volume, the highest protein amount, and the total protein is the highest. But this is uh, in total, right? Not a specific activity yet. You see, the specific activity is the lowest because it's a lot of protein. Okay, when you do slowly, when you do the protein purification step, you will see the specific activity increasing. So you can see immunoaffinity having the highest specific enzyme activity. All right. So and then, but you will see the volume is less, the protein also less, because you will lose a lot of protein. You're not lost protein because of the amount of protein that you need is very low in the extract. So meaning you have already removed out the others. So the protein amount that you need is only around 1.8. This is the protein interest that you want. And the total activity is 275. Okay. But your specific activity is the highest because that is the enzyme that you want, for example. Okay. So can you understand this? You get what I mean from this protein purification table? Guys, can you imagine what I'm saying just now?
Can, doctor?
Okay, all right. But as I said, if you don't do the experiment, it will not as clear as what I explained it to you. Because you see, there's a 5 different chromatography, including the screw extract. It will not enough for 3 hours. Even if you want to do, it will take like 3 to 4 weeks because it has a several steps. So since this is not an enzymology class, so I just give you the idea only. Okay. So first, protein solubility. So you need to know when you handle protein, so you know that they have an isoelectric point. So you can increase the solubility by adding salt. So you know the protein can be soluble, right, in water. So in order for you to increase the solubility through the ionic strength, you can add salt. So we call this salting in. Okay, so it will cause the salt and the protein to undergo electrostatic attraction. Okay, and then when you increase slowly, so then later the solubility of protein become decrease later, right? So that is, we call it salting out. So meaning now the salt is too much, so that the protein cannot be soluble with the salt solution. So now it tends to be precipitate. So there is several salt that you can use, but usually we use ammonium sulfate precipitation. Okay, so this is just a schematic structure to give you the idea. So due to the hydrate shell, so the orange is the protein, so blue is the salt, right? So when you increase slowly, we call it salting in. So the protein will tend to soluble, right, in the water. But at a certain point, there will be a reaction with the protein and the salt. So later it will cause the protein to be salting out, we call it, right? So the salt will not be able to bind. So now the protein will be precipitate. It become like a cloud whitish. So you can centrifuge and you can collect the pellet. So the solution is basically all the salts. So now you can separate the protein out from the salt solution. This is how we do a protein precipitation, by slowly increasing the salt. All right. So and then we can do the protein separation, as I mentioned earlier, through by size. Okay, you can do ultrafiltration, or you can do centrifugation, and you can also do a size exclusion chromatography. Okay, so you can separate them through size. Okay, this is to give you the idea. Gel filtration, so you have this is the matrix. You can put into your sample on top of the matrix, and it will slowly go down through the matrix. So since the matrix have a pores, right, so the big protein will not be able to enter the pore size. So the smaller protein will be able to enter the pores. So that's why the smaller will come out later. The big molecule or the big protein will not be able to enter the pores. They will go out first. So that's why when you do a fraction collection, you will get the bigger size first, followed by the middle size and the smaller size. So this is the principle of gel filtration. And you can also separate through electrical charge. Okay, either you do the ion exchange just now. So we have matrix. For example, if your matrix is positive, your protein that is negative will bind. So if it's the same charge, it will come out first. So that is the principle. And you can also separate it using gel electrophoresis, right? So you have a gel, and then you supply an electric. So you have a negative electrode on the upper part and the positive electrode at the bottom. So you have to add a detergent in order for the protein to separate into within this gel, right? So since, as I said, this is not a enzymology course, just give you the idea how protein can be separated. Okay, and this is the ion exchange chromatography. So as I mentioned, depending on the matrix that you use, if your matrix is negatively charged, so the positive ion protein will bind, and the negatively charged protein will come out first. So when you do a collection, so the negative will come out first, and then lastly the one that is bound to the matrix will come out last. Okay, and affinity, so you have a matrix, and then you design a matrix that have a specific ligand, usually antibody. So this antibody, you design that can bind to your protein of interest. Okay, so when you put in your sample, so your protein will bind to the ligand. The others will go out, will be removed out. Only the protein that bind or the target will stay in the column. Once you have flushed out all the unwanted protein, you can elute it. You can elute the protein that bind to the specific ligand, then you get a pure protein. Okay, so this is what we call it affinity chromatography. And then lastly is the protein purification using a jet electrophoresis. So there is two types. Basically the base is the same. Sodium SDS is the sodium dodecyl sulfate, is the detergent. Uh, we link it with the gel, polyacrylamide gel. So we call it SDS based gel. So this will help you to separate according to the size. Similarly to the 2D gel, but this one you can separate into another dimension, isoelectric focusing, where you can separate the protein according to their pH as well as to their size. Okay, so this is only 1D, or we call it SDS page. Only you can separate from the higher size molecular weight to smaller molecular weight. Okay, so this is how they migrate from negative to positive electrode. Or you can do 2D gel, where first you separate the protein based on their PI, their net charge, pH. Once you separate it according to their pH, then you run the gel SDS page, so that you will get a spot or protein like this. Instead of band, you get a spot, right? Protein spot. So you can see that it being separated according to size and pH. All right. So why study protein? So basically, most protein will involve in the human physiological function. So it will help us to study or to help in medicine. Okay. So if you look in the some example is sickle cell anemia. So it is actually due to mutation of the beta chain in the hemoglobin. So if beta chain is mutated, so basically the sick amino acid residue from glutamic acid is changed to valine. Alright, so it will cause the normal biconcave disc of red blood cell to become elongated sickle shape. Sorry, so that is cause the patient who have sickle cell will have anemic and decrease in their hemoglobin content. So usually they show frequent illness, right? So this is how the sickle cell or red blood cell looks like under the microscope. Okay, and there are also other diseases that related to protein folding. Okay, due to the abnormalities of protein folding, so it may result of loss of function. Okay, for example, Alzheimer's disease. So this is common. Okay, so beta amyloid peptide derived from this amyloid precursor is misfolded. Okay, beta amyloid peptide will be accumulate in the human neural tissue. Okay, so this is when we get older, this beta amyloid become misfold. There are others. I think P53 is common in cancer. Alright, P53 will prevent cell with damaged DNA from dividing. Okay, so one class of P53 mutation will lead to misfolding. Okay, the misfolded protein will unstable and destroyed. Okay, so there are others example. Okay, you can read out. So basically all are involve the misfolding of the protein. Okay, all right. I finished it very early at 11, 1 hour earlier. So any question? Did you receive?
No.
Yeah, do you see the tutorial 3 in the Petrobras?
Yes.
Okay. Yeah. So I think it's straightforward question. So can you be able to do it by next Friday and we discuss tutorial 3? Is it possible?
Yes.
Okay, so I will try my best to get the class 1.3 in the morning at 10. At 10, right?
Yep.
Okay, I will try to book next week at 10, bilik seminar 1.3 to discuss your tutorial 3. And then the following week is your test 1, which I informed Inshira that we have to change the time because the place 1.3 on 17 there is a another event on Friday. So they need to use 1.3 on 17. So I have no choice. We have to do it during your lab session. So the test will be in bilik seminar 1.3, right? During your lab session, 15 November. Okay, instead of having a practical class, you will have your test 1. So meaning week 6, there will be no practical session. Okay. I did inform, right? The topics for test 1. It will be covered your topic 1, 2, 3 until today. Plus your practical 1, 2. 3 will be not covered in test 1. Okay. Do you have any question? Have you take your attendance in the Putra Blast?
Yes.
Okay, so that is an evidence. So later on Monday, I will ask you to sign twice because it's considered that 3 hours even though I finish up early. Okay, so I need you to sign twice later on next Monday as a proof. But I will check whether you have tick or not today's attendance. Okay, if you forgot, please inform me that you forgot, that I will consider that you are attending the class, today's class, right? Okay, if you have no other questions or any issue, maybe we can dismiss early. Any other issue you want to discuss?
No, doctor.
Okay, so I will see if I can get this 2 recorded lecture. If I manage to get it, I will share to you in the Putra Blast. Alright? Okay? Okay, doctor.
Okay.
Okay. Alright, see you on Monday and have a nice weekend. Alright.
Thank you, doctor.
Thank you, doctor.
Thank you, doctor.
Thank you, Doctor.
Thank you,
Thank you, doctor.
Thank you.
Thank you.
Thank you, doctor.